Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health.
I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Raffaele Teperino. This is an amazing episode. When we talk about conception, we usually just think about the mom, right? The maternal side of things. But Raffaele has actually been digging into the role of epigenetic material from sperm and more of that father or parental side of conception. It's something, again, that we just easily breeze over, but is as equally important. So we talk a little bit about Raffaele's background and how he really explores reproductive fitness within his research. We also talk about how epigenetic information can be passed through the sperm and eggs and what his recent research has uncovered about this. I even talk with him about what it would look like to really optimize conception from a male standpoint, but even female standpoint, and the ways which we can intervene during early stages of life to really ensure that there are better outcomes. We dive a little bit into how this can have a huge effect on public health, even looking into any red flags or signs that doctors or parents should look at for and during pregnancy, and how early life risk like childhood obesity, diabetes can even start before birth. We end by him giving one piece of advice based on his research. He actually ends up giving us two, which was amazing, and obviously ending with what excites him the most about the future of epigenetics and its really potential to change how we think about health, especially as it relates to conception, which is extremely important in this day and age. A little bit about my guest, Dr. Teperino is leading the environmental epigenetics group at Helmholtz, Munich, focusing on epigenetic inheritance of complex traits. He earned a PhD in molecular endocrinology and conducted postdoctoral doctoral research specializing in development, epigenetics, and metabolic physiology. He has over 36 scientific papers in top-tier journals, and he's been acknowledged by the European Commission as an expert in personalized medicine, and his research really advances knowledge in epigenetic inheritance and metabolic physiology, focusing on these insights, what we're learning, and how we can actually turn these into treatments for metabolic diseases, which is extremely important in my eyes. We need to make this as applicable as possible. I hope you all enjoy. And now for my guest, Dr. Raffaele Teperino. Welcome to the Everything Epigenetics podcast, Raffaele. I'm excited to have you here today.
Hi, Hannah. Thank you very much for having me to your very successful podcast series.
Yeah, I gave a nice introduction to you at the beginning there, but just to reiterate, I'm extremely excited to talk with you, particularly about your work in epigenetics and the transfer of that epigenetic material from sperm to oocyte during fertilization. And you've recently more discovered that paternal health, which I feel is not as talked about as maternal health, at conception is a distinct risk factor for childhood obesity and diabetes. So we're going to get into all of that today, but I'm going to walk before I run and want to ask you, what first got you interested in studying epigenetics? I know everyone's story is a little bit differently, but was there a particular aha moment? Was it a longer journey?
Well, my story comes from my education, right? At the time of university where I studied biotechnology, it was the time of gene and association studies. There was a boom of genetics, right? And we all, as a students, we already hoped that genetics would solve complex disease. So with genetics, we would understand complex disease pathogenesis. Well, indeed, genetics has allowed us to advance a lot. But then I started my PhD and I started studying type 2 diabetes, which is one of these complex disease that was thought to be genetic. And I was studying risk of getting type 2 diabetes in first-degree relatives of diabetic patients. And my PhD essentially ended up discovering that their risk is independent from their genetic blueprint. And so I said, there must be something on top of it that makes a difference, right? And that was the moment where I started looking into epigenetics and see if I could find kind of a better answer to this varied in the pathogenesis of diabetes and other complex diseases.
Mm-hmm. Yeah, that's super interesting, right? You're asking what else is out there. There has to be something more to this. You would absolutely, I mean, I think anyone, even more of like a general consumer, right? Someone who's maybe not in this realm of the scientific world as we are, would think, oh yes, my ancestors, my mom, my grandma, etc., has type 2 diabetes. I'm going to get it, right? We fall rather victim almost to our genetic makeup. And again, I think that's the power of epigenetics, is that it's in your own hands to an extent for some particular outcomes and diseases.
Yeah, the good thing is that, despite being somewhat determined by your ancestors somehow, right? They're still flexible and reversible. So you can still work against it. You can still try to fix it somehow.
Yeah, exactly. With and we'll talk about that later on as we get more into the podcast, but lifestyle factors, right? Supplements, medications, procedural-based therapies. I think that's what everyone's interested in, is what can I actually do to start to make that change, and how can we measure it and affect it? One thing I want to ask you, Raffaele, as well, and what I already mentioned is the most exciting part of your research to me, is really how you've been exploring how epigenetics really ties into this idea of reproductive fitness. Now, can you walk us through what that connection looks like? And maybe for those who aren't as familiar with reproductive fitness, you can define that term first.
Yeah, sure. That's a very interesting question. Actually, I like to divide reproduction into two sections. One is reproductive capacity, which is the capacity we all should have to give birth, to produce, quote unquote, a child, right? So that's our birth capacity. Reproductive fitness, it's slightly different from capacity, because on top of it, it has. So we need to be able to give birth, to produce a child, and our reproduction is fit when you are able to produce a healthy child. So when we are able to produce healthy pregnancy and healthy child. And this is essentially where epigenetics ties in, right? So epigenetics make our reproductive capacity more or less fit in a way. And this fitness is heavily influenced by the environment we go through, by the environmental challenges we see during our lifetime, by our lifestyle choices, our nutrition, our many, many different things that can impact on our epigenome. And our focus is in particular on how mostly dietary choices impact our epigenome. And our two most recent studies were on how what we eat impact our epigenome, and how this impacts again our reproductive fitness. And on a different study, we also try to understand not what we eat, but when we eat, whether when we eat as an influence on our epigenome and our reproductive fitness, and then in the end, how it impacts our offspring long-term health.
And when you say when we eat, can you elaborate a little bit more? Is that just timing of when you eat, or how much you eat? What do you mean by that?
Well, and the idea was to understand whether there's a hype on nutrition at the moment, right? Where there are studies showing that it doesn't matter where you eat, but if you eat it in a defined time window, then the health consequences is going to be, it's going to be more or less dangerous for your metabolic physiology and for your overall health consequences. And this is essentially what we try to reproduce in the mouse, because what we do is mostly research on animals, right? So we mostly study animal models.
Got it. Understood. So the time-restricted feeding and the intermittent fasting, those type of models. Okay, perfect. No, I love how you broke that up. So you said, all right, well, we really define this in two ways. You have your reproductive capacity, which is just being able to give birth, to reproduce in this world, to continue, our life. And then you have your reproductive fitness, which is that healthy pregnancy, that healthy child, that healthy birth. Can you define healthy, right? Like, what are you actually looking for? Is that just reproductive fitness in terms of offspring that has, decreased or lesser chance of disease later on in life? Is that how you're defining that?
Exactly.
Okay.
Yeah, exactly.
Perfect. Yeah, go ahead. Did you have something else to add?
I was saying that, on one side, this offspring with a reduced risk of complex disease or like unhealthy life, but also at the same time, pregnancies which have a low risk of being complex, right? So develop with healthy placenta and functional placentas, which are, these are all things that are connected to each other, right? So
Yeah, it's also interconnected. It's hard to really separate them at some point too.
Yeah.
So, and I guess this leads into my next question. I find it very fascinating that the sperm and the egg carry way more than just DNA. I think we know they do, but when you're learning about it, even through, middle school up until high school, we're really taught, we get 50% of our DNA from parent 1, 50% from parent 2, but we really don't talk about anything else, right? They're also passing this epigenetic information to us. And I absolutely find it fascinating, the epigenetic transgenerational inheritance. So can you share anything that your research has uncovered about this? Is there a way we can start to look at these epigenetic modifications that our parents are passing off to us, or even grandparents, right?
Yeah, sure. Actually, that's the most exciting part of my everyday life, in a way. So, yeah, as you said, we're starting understanding that sperm and eggs, they don't only transfer 50% of the genetic material, right? So they don't only contribute 50% of what we are as adult individuals, right? What they do, and we mostly focus on sperm, they also record lifestyle before conception and then transform, in a way, this lifestyle experience into small molecules, which are meaningful for the next generation. And these small molecules are so-called epigenetic mediators, and they get transferred to the next generation. Actually, we've been the first one to show this transfer, like to be able to see these molecules going from sperm to eggs. And once they are in the fertilized egg, these molecules have the potential to steer development from the beginning, right? So and then kind of address the next generation towards different life trajectories, in a way. And what we have essentially discovered, which was very exciting for us, is that sperm cells are thought to be lazy bumps, right? So they are transcriptionally inert, they're translationally inert. Once they leave the testis and enter the maturation phase in the epididymis, they're thought to do essentially nothing but swimming and reaching the egg and fertilize and transfer 50% genetic material. What we've discovered instead is that this phase of sperm maturation is a very sensitive phase to environmental challenges. And maturing spermatozoa can actually sense the environment and mount a so-called an epigenetic response that is then transferred to the next generation and influence offspring life trajectories. And this response, essentially the one we found, this response starts from a separate DNA molecule, which is not the nuclear one, but it's the mitochondrial DNA. And now there's a whole discussion on whether mitochondrial DNA is only, there's discussion that mitochondrial DNA is only maternally inherited, right? And that fathers do not contribute any mitochondrial DNA to the next generation, which is true. But what we found is that fathers contribute products of this mitochondrial DNA, which have the potential, and actually do, influence the activity of maternal mitochondrial DNA in the next generation. So it's a new way of parental communication at conception, if you wanna put it like this, right? So this epigenetic material coming from sperm modulates specific responses, specific responses into the next generation, and then design trajectories. So this was the most exciting finding to my point of view. And interestingly, our results, they aligned very well with our results in mice. They aligned very well with results we obtained in humans, and also with a more recent study from a colleague in Sweden, that they found the exact same response in a very defined human cohort upon a different lifestyle intervention, suggesting or demonstrating, if you want, that this epigenetic machinery is the one responsible for responding to specific environmental challenges in mature spermatozoa.
Okay. Yeah, very interesting. Like you said, the sperm has been thought of as lazy. It just passes off that 50% DNA and we're done. But what you've learned with your research is, sure, maybe you don't get that parental sperm transferring all into the mitochondrial DNA or actual material, but there are products that we do see from that sperm. So there is a very clear connection there. Now I have to ask you a personal question as well. Do you have any children? 2 children? Okay, perfect. I don't have any yet, but I definitely want some in the future. So when you think about your research, does that affect kind of how you live your life, especially from like a reproductive standpoint? So that's the first question. And then my second follow-up, and we can come back to this, is what's the time frame, right? When people are starting to try and conceive. I think I've heard around 3 months, right? Around before we start to conceive, stop drinking alcohol, stop all of those bad habits, try and live that better lifestyle we were talking about. So I'd love to hear your thoughts on both of those.
Well, I'll give you a personal answer first. Well, I mean, I had children before we made this discovery. So now it kind of changed the way I look at my children in a way, right? Because I try to live healthy, and I'm leanish, but I don't think I'm the healthiest person on earth, right?
Sure.
So I keep an eye on my children to see if they might have inherited some of my waste epigenetic signal. But I can't see signs of that yet. But on a more serious side, yeah, I think that's sort of what you said is correct. So it's almost 3 months for a full spermatogenesis cycle in humans. So it's 10 to 12 weeks that humans take to regenerate a whole sperm pool. So if you want to if you want to try to provide the healthiest sperm at conception, my suggestion would be for fathers to start thinking at their own health. I mean, they should constantly think at their own health, but particularly when they start trying to have children, think of their own health kind of in an interval which is 10 to 12 weeks before conception. And being healthy means, reduce sugar intake, reduce fat intake, take supplements that might reduce mitochondrial stress, so antioxidants, specific, increase the consumption of specific vegetables that are known as epigenetic promoter, in a way that increase epigenetic robustness. And yeah, these are all things that might help might help offspring health. Unfortunately, we still don't have specific biomarkers for this, and we still haven't run a proper risk stratification study to understand what exactly makes a father at risk of giving birth to a child which has higher risk of developing obesity or insulin resistance and other complex diseases. But what we for sure know is that if you improve your metabolic health, this is what we found in mice, right? So in mice, if we make mice overweight at conception, we get all the health consequences the next generation. But if this other way is reversed before conception on a new pool of sperm, then there's no phenotype anymore in the next generation. That means that we can reverse this diet-induced phenotypes.
Mm-hmm. Yeah, I mean, absolutely. And that was my next question. You really already answered it was, hey, I want to talk about all the dads out there for a minute, right? How does a father's health at conception influence their child's health? And I think it's everything, right? And right now, since we don't have a biomarker, since we don't have a risk stratification to really give people an exact plan, we can definitely use what we already know from a healthy lifestyle, right? Like you said, including more of those epi-nutrients in the diet, making sure we have a basic physical fitness routine. We're not smoking, not consuming alcohol, watching our insulin sensitivity, getting really great sleep, and trying to have low stress. So that's a long list, but I think it's the basics. And I think if you cover that, then like you mentioned, you're going to have better offspring. You did mention the biomarker and the risk stratification test. Is that of any interest to you and your work in your lab? Is that something you want to create maybe someday or eventually commercialize?
That's what we're trying to do at the moment. We're trying to see if the molecules we identified can be used as biomarkers.
Okay.
That's what we're trying to do. And of course, to do this, we can't use mice, right? So we need to scale up and try to use humans. And this is what we're trying to do. And in the future, hopefully, that's the goal, right? The goal is to provide a full list of potential epigenetic biomarkers for reproductive fitness, which goes into the next generation, so intergenerational reproductive fitness.
Yeah, definitely. And when we back up and again talk about more of this larger picture, you've done a lot of great work as well in the preventative medicine space. I know the European Commission has acknowledged that you're an expert in the personalized medicine. Again, focusing on the epigenetic inheritance, the metabolic physiology, and translating that for treatments of metabolic disease. But what do you think, outside of getting a test on the market, from that public health perspective, that we should be doing to integrate epigenetics into preventative medicine? Is there anything else from, sure, the lifestyle factors and what have you in integrating this test, but what else are you wanting to see?
Well, from the test point of view, I think especially for paternal, especially for sperm health, that should be easy enough, right? So once we have identified biomarker, I hope to see there's a routine sperm test, so sperm epigenetic test. For example, right? So if now a couple goes to a doctor and says and express the willingness of having a baby, right? So the doctor attention goes to the mother, right? It goes to the woman, and they start saying, okay, then you need to take pharmacy, you need to stop drinking, you need to stop smoking, blah blah blah.
Sure.
And the father is completely overlooked. And I think that needs to change, right? That needs to change, and we have sufficient amount of data that could push health policy to change this, right? And then the idea is so once we have, as I said, once we have identified biomarkers, the idea is that they'll run an epigenetic test, a routine epigenetic test on fathers to be, and at least to inform them of how healthy they get at conception, right? And what kind of signals they are transferring to the next generation. And, once we have enough knowledge, the goal is to then instruct fathers to be to revert their sperm epigenome or correct or mitigate their sperm epigenome in a way that the inborn risk of their offspring is going to be as low as possible.
Mm-hmm. Mm-hmm. Definitely. Yeah. And, I even a little personal story from myself as well. I just figured out that my grandmother was smoking cigarettes when she was pregnant with my mom. I had no idea. I was talking to my mother about, epigenetics and the work I was doing. Right. And again, we're talking about kind of what my maternal side had gone through, but I didn't even think to ask, right, my dad or my father or kind of any of my grandfathers too. So I think just changing and shifting that way that we think about conception and pregnancy, right? It definitely takes two. We're focused on the mother a lot, which I think we still should be, but we definitely need to bring in more of that parental side into the research, which is why I love what you're studying. I'm also thinking about all of the other applications that this can really affect. Do you see this also entering the space of, IVF essentially as well, or even people who want to become sperm donors, right? Almost using some type of test to rank the reproductive fitness levels and to give maybe females more insight into choosing how they want to proceed with conception and pregnancy.
Yeah, we're getting to a dangerous level there. But
We are.
yeah, but definitely, that could be. But as I said, we need to have solid basis before we can make any conclusion on this. But definitely, right? That could be an avenue, especially for couples undergoing IVF. And this is that this numbers are going high up and up, right? So and IVF brings its own risk, right? So it has its own problems. It brings its own risk to the next generation. So getting to the IVF as healthy as possible and with the best epigenome as possible, then at least you can mitigate part of this risk. And another option, another dream, which is quite complicated at the moment, but it could be to do some epigenetic tests on embryos. Include epigenetic test on pre-implantation diagnosis, right? You can select the best embryos for implantation and next generation.
Yeah, definitely. I want to go ahead and talk about a little bit more and dive in a little bit more into more of your recent research that's highlighted how your early life risk, like childhood obesity and diabetes, can even start before birth. So can you tell us a little bit more about that research?
Sure. Yeah, as I said, what we found out is that using a group of more than 3,000 families, current families which are highly characterized. So the parents are highly characterized at conception, and the offspring are longitudinally followed up until 18 years of age. What we discovered is that by analyzing this data, having a father which is overweight at conception doubles offspring obesity risk. And doubles offspring obesity risk can increase their risk of developing insulin resistance as well. If the father is obese at conception, this risk increases fivefold over the general population, right? So it's a huge impact. And this is independent from maternal BMI at conception, and it's also independent from genetic risk score and shared lifestyle. So we looked at the genetic risk score in the family, and we looked at the lifestyle of the family. So, screen time, food habits, eating habits, sport, and so on. And this is all, so there's a predetermined risk that only comes from paternal BMI at conception.
Sure.
And when we then translated this model into mice, what we found is that if you make a mouse overweight at conception, the offspring, so 30% of these offspring develop glucose intolerance, insulin resistance, and go towards type 2 diabetes. So just only 30%. So this phenotype is partial penetrant, right? You don't see the whole generation in the whole population. And what we interestingly found out using sophisticated single embryo profiling. So we essentially took sperm from these mice, made an IVF, and started single very early embryos right after zygotic genome activation, so in the first phases of development. What we found is that already in these very early phases, you can distinguish two different populations, which are in the ratio of 30 to 70, so resembling the 30% penetrance that we see in adult offspring, and the 30%, this meta population has signatures that are connected to late onset metabolic disorders. So actually, the trajectories of this adult offspring were already there one day after conception, after fertilization, which opens up a window of intervention, right? So as we were saying before, right? Opens up a window of intervention, especially for IVF, where you can study pre-implantation embryos and kind of rank them, kind of stratify the risk of giving rise to offspring with metabolic disorders, for example.
Mhm. Gotcha. So just so I understand you correctly, there was this human study, right? They had about 3,000 families that they looked at longitudinally, correct? And then you have the males you characterize as overweight. They had a 2 times diabetes risk passed on to their children, right? The next generation.
Exactly.
Okay,
Yeah.
perfect. And then you had the obese men as well, who were 5 times more likely to pass off metabolic disease, type 2 diabetes risk to their children. So that was independent of any maternal factors too. And then you were wanting to kind of study this process in mice, right? So that's what you all did. And
Yes.
you saw from the overweight category of about 30% of signatures being passed on in mice, right? And those were the signatures that may have told you more information for late onset of metabolic disease. Did I get all of that correct?
Exactly. Yeah, that's
Okay,
correct. That's
perfect.
correct.
I was trying to follow that. And then, like you said, yeah, there's that window that you have open. There's timing there where you can start to study the in the pre-implementation phase, those embryos, right? To say what risk are we carrying on. So that's just wildly fascinating. Are there any studies that you know that are dependent with maternal information as well, obviously, to see kind of the effects of both?
Yeah, there are studies. There's actually one study from like almost 10 years ago that show that they use a very clean system because they only generate offspring via IVF, right? So they control for any confounding factor. So they took sperm and oocyte from obese male and female. They did IVF, and then in mice you can implant these embryos in a different mother, and then once this healthy mother gives birth, then you can even cross foster the offspring. So let them be lactated by another mother. So essentially, the signal they get, it's only coming from the gamete. And what they showed is that even in this setting, the offspring coming from mothers that were obese before conception, they have a higher risk of developing obesity and diabetes upon a second diet hit, right? So they don't have an inborn risk, but when they're fed a high-fat diet, so they're put on environmental stress, like on a diet hit, they're more likely to shoot out and develop obesity and type 2 diabetes.
Okay, so that's it. Yeah, go ahead.
Well, if you have them on a normal diet, they're perfectly normal. But as soon as you stress them with a diet, then the phenotype kicks out. It's like they have a pre-memory, right? So they're primed to a certain response.
Yeah, and that's super interesting as well. If you're looking at it from more of the IVF landscape, you have sperm and egg from obese mother and father, and then you actually implant it into a healthy mother. You're saying,
exactly.
Sure, they're fine on a healthy diet, but once you switch over to that unhealthy diet, you're actually going to start to see some of those phenotypes and outcomes appear. And then there was another study more recently that actually was just published on epigenetics. It was like, I forget the name of it. I didn't read it all. I briefly skimmed through a couple news outlets that wrote about it, about kind of our epigenetic memory and why it's actually so hard for some of us to maybe lose weight or keep the weight off. It's because our cells do remember at one point we may have been, not as metabolically optimized, right, as we once were. And our cells actually remember that information. And that's probably a big reason as to why it's hard to shed weight or keep that weight off once we reach a certain point.
Yeah, it's true. That's true. That's actually a study, like another landmark study in this epigenetic scenario. The only thing I like to point out about this study is that what we know about epigenetic signals and mechanisms is that they are reversible, because every cell has a proper machinery to set up an epigenetic landscape and remove it, right? And most of these enzymes, they respond to metabolites, right? They work with cofactors. So they sense the level of I don't know, methionine, a precursor of acetate precursors. So if you modulate, so they respond to the metabolic environment. But what we don't know is what's the timing of this reversibility, right? So how long you need to wait before you see these epigenetic marks reverse and come back to normal. And in this study where they said that once you're obese, even though you try to get lean, you never become lean because your cells remember you've been obese. They only looked at a defined window, right? So they did a lifestyle intervention for a defined window. They didn't see reversal of phenotypes and reversal of epigenetic marks. But they didn't extend it. They didn't chronic lifestyle intervention to see if eventually at certain point this memory could be wiped out. And
how long was it that how long did they do the intervention? Do you remember? I don't remember off the top of my head.
Yeah, me neither. I think it was about 2 months.
I was going to say 2 or 3 months.
Yeah, I think it was about 2 months. But no one knows the timing.
Yeah, that's the issue here too, because again, that's the exciting part about epigenetics, right? That's why it's this hot, sexy topic right now. We can change it. We can do something about it. But then it's, okay, well, what do you do to change it? And then how long does it take? And I think that's that's the biggest question in the space right now. And there's a ton of different factors in play. So how are we able to isolate them? And that's why I think the work you're doing is very interesting, because you're starting way back at the beginning, and you are able to isolate it to an extent and really start to point back to the starting point in a way. So that's super, super interesting. Talking about maybe getting into some uncharted territory. One question I want to ask you, Raffaele, is we've been talking about things you can do, right? Especially from a paternal side, waiting 10 to 12 weeks to regenerate your sperm pool, kind of things you can do from a lifestyle factor. But are there any red flags or signs that parents should look out for when they start to think about conception and pregnancy, or even, you can think about it from more of a scientific research kind of doctor standpoint as well, just when you think of it in the sense of epigenetics?
Wow, that's an interesting question, because we get into a kind of a cloudy space, right? Mm-hmm. So So my instinct answer would be, yeah, there are many red flags that we should take care of, right? But they're all interconnected, right? So it's easy to say, yeah, look for your BMI, look for your whatever blood pressure, look for your blood glucose, look for these are all red flags, right? So all red flags from maternal and paternal perspective. These are all these are all very unspecific, right? So these are all things that should be looked at. And that's a matter for which reason, right? This is something that we all should take care of. For specific epigenetic risk, or what we like to call intergenerational epigenetic inheritance, I think we're still far from identifying specific red flags. Mm-hmm. Once we have them, I think it's relatively easy to go from identification to usage of these red flags, right? So from identifying them to use them to kind of preventive strategies and reduce prenatal risk. And that's the goal in the end, right?
Yeah, that makes sense. I think that's the perfect way to answer that question. That's a hard question. There are many red flags, but like you said, we should be doing the basic things that we know are going to give us an increased risk in any type of metabolic disease, the blood pressure, the BMI, the blood glucose levels, right? I actually have a CGM on right now that I'm playing with. You can kind of see it on my arm.
I'm not
diabetic, but again, just learning a little bit more about my body, and I think empowering myself with that knowledge to help me make better lifestyle choices. I would love to see in the future, definitely one epigenetic tests that's showing some type of that transgenerational epigenetic inheritance. I'm most familiar with Dr. Michael Skinner's work out of Washington University. And I remember listening to his lecture about this subject 2.5, 3 years ago. And I was like, why haven't we been talking about this for years, right? Going back to the public health standpoint there, policymakers don't look into different generations to even make policies as it relates to our environmental factors. So even being able to start with us right now and really start to think about those future generations, I think is really important.
Right.
Yeah.
And we should, since we're talking about reproductive fitness, I think we should also move away from germ cell only, right? So we know that germ cell they come with an accompanying fluid, which is the seminal plasma, and this also has significant impact and surprisingly significant impact on conception, on pregnancy outcomes, pregnancy health, and offspring health. And this is another thing that we should take into account, especially now with the increasing amount of IVF that we do. And if there's one thing we do with IVF, we just get rid of this fluid, right? Because we isolate germ cells and we use them pure without something that may be useful.
Yeah. Can you, for the listeners who aren't familiar with those terms, can you define germ cell and then define the seminal plasma as well?
Yeah. So germ cell are where we were talking before, right? So sperm and eggs. These are the gametes, the cells that make a new organism. And seminal fluid is a fluid that it's produced by the prostate and seminal vesicles in the male tract. And this fluid accompanies germ cells into the female tract. And it was start, until a few years ago, it was start to only kind of push the germ cells up the female tract into the oviduct until they find an egg that can be fertilized. But now it has been discovered to be an endocrine hub. So it contains hormones. And these hormones are important to prime the female tract to the conceptus, right? So they are important to make the female tract ready to accept the blastocyst. And also what we have recently discovered is that components of this seminal fluid are also important for placentation, so for formation of placenta, the most important fetal maternal interface organ. And so this components of seminal fluid are important for producing placenta, for producing a healthy placenta. Yeah. And you know that placental problems then lead to health problems in the next generation.
Sure. Yeah. And you're saying that seminal plasma is not passed on, obviously, when you're doing IVF, correct? Right.
Yeah,
right.
Exactly.
So it could be one of the reasons why we see differences in reproductive health versus, IVF and not IVF. But I think,
exactly.
a lot of different interesting ways you can go there. There's so many things I want to talk to you about, but trying to keep it focused here. And we're getting to the end of the podcast, Raffaele. I have a couple more questions for you. I guess the main last one that I have for you is we'll do two more. If you could give listeners one piece of advice based on your research, what would it be? We've talked, I think, generally about some things, but if you could just pick one thing, what would that one thing be?
I'll give two.
Oh yes, perfect.
The first one is to try to get at conception as healthy as possible, right? As we said before, right? So try to take care of your health. And taking care of your health means start eating well. So prefer polyunsaturated fatty acids to saturated fatty acids. Reduce sugar intake. Reduce or eliminate alcohol intake. Don't smoke. Do normal exercise. Sleep well. And these are easy things that can make your life better and your son or better, so your next generation lives better. So you're saving, quote unquote, saving two lives in one shot. The second thing is more of a hope, and that's also what we were discussing before, is that don't feel guilty if your lifestyle hasn't been exactly the best one until now, because your epigenome is reversible. So you can always get back to your lifestyle choice. You always have a chance to make it better for the next generation. So there's always time to act against it, to work for it.
Yeah, I like that. Yeah, it's that little piece of hope. Again, it's understanding that you can change, you can still make those healthy decisions. All right, last question here as it relates to your particular work. What excites you the most about the future of epigenetics and its potential just to change the way we think about health? This could be something we haven't even talked about today. This could be something as it relates to your research, but why do you get excited about this?
I think epigenetics has the potential. So what excites me about epigenetics is that it has the potential to explain a lot that we still don't know. And one question we're running against now, and we're trying to understand is, well, we were broadly interested in complex diseases, right? We've been talking about metabolic disorders and the risk of metabolic disorders, but there's one statistics that kind of kills me when I see it, and that's the race, and that's the exponential race in neurodevelopmental disorders. And I think, and this is so, our research is going more and more into this direction. So we're trying to understand how neurodevelopmental disorders are epigenetically programmed, and how they can be epigenetically programmed by us as parents. And I think we are at a point that we are identifying some key mechanisms. And so what excites me is that I'm gaining more and more, I'm becoming more and more conscious that at certain level we might be able to use epigenetic mechanism to prevent some of these complex diseases. And the more we understand, the closer we get to a light, in a metaphoric way.
Yeah, I love that. I mean, I definitely want to see it in my lifetime, right? Where you go in the doctor's office, it's a finger prick, blood spot card, or saliva tube, better yet, and you're able to just understand your risk for everything, right? And then change that risk. But awesome. Well, Raffaele, I have another question I ask everyone at the end of my podcast. It is, if you could be any animal in the world, what would you be and why?
Any animal?
Yes, it's the hardest question that you've answered today.
Well, it's not a weird question, but I give a weird answer. If I could choose, I would be a mosquito. Like, it's more mosquito.
Oh,
and the reason is that because I want to be everywhere without everyone noticing me.
Okay. Interesting. No one has said mosquito yet. I don't think anyone has even chosen like a type of bug or insect, but I like it. Be a fly on the wall and be able to go everywhere.
Exactly.
Perfect. Well, we've come to the end of this amazing podcast interview. If anyone wants to get a hold of you or, wants to find a little bit more about your work, where can they find you?
They can find me on social media. So you can find me on Twitter, on Instagram, on LinkedIn, and also at my institutional email address. I'm a principal investigator at Helmholtz Munich in Germany. And if anyone is interested, just get in touch with me. I'm happy to discuss anything.
Perfect. Yeah, everyone, if you're interested, just Google his name. He'll pop up. You'll see him all around Google and Google Scholar. So I really appreciate your time, Raffaele. Thanks, everyone, for joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more about how.
Thank you, Hannah.
You're welcome.